Next, we move on to specification 4, which asks us to discuss potential advantages and risks of genetic modification. Genetic modification offers several important advantages, especially in agriculture and biotechnology. One major benefit is that it reduces the need for chemical herbicides and pesticides, making farming more environmentally friendly, less time-consuming, and more cost-effective. It also increases crop yields because genetically modified plants are better protected from pests and face less competition from weeds for water, nutrients, and sunlight. In addition, genetically modified bacteria can reproduce rapidly and produce large quantities of useful substances, such as insulin, in a short time. There are also fewer ethical concerns when using microorganisms like bacteria compared to animals, and because all living organisms share the same genetic code, genes can be transferred successfully between different species. However, genetic modification also comes with several risks and disadvantages. One major issue is cost, as GM seeds are often expensive, making them less accessible for small or low-income farmers. There is also increased dependence on specific herbicides, which are usually produced by the same companies that sell the seeds. Another concern is that modified genes may spread to wild plants through pollination, which could lead to the development of herbicide-resistant weeds. Genetic modification can also reduce biodiversity, as fewer plant species may survive in areas where GM crops are grown, which can negatively affect insects, birds, and the overall ecosystem. In some cases, GM plants may not grow as effectively as non-GM plants, and there is also a risk that modified genes in bacteria could be transferred to other bacteria, leading to unpredictable outcomes. As you can see in the diagram, the process of agricultural biotechnology begins with identifying and extracting a useful gene from a source organism, such as bacteria. This gene is then modified and inserted into plant cells. These modified cells are grown in controlled conditions to develop into small plantlets, which are later grown into full plants. Through further breeding, the desired trait becomes stable and is passed on to future generations. This process allows scientists to develop crops with useful traits such as pest resistance, herbicide tolerance, and improved nutritional value. So that brings us to the end of Unit 18.2 on genetic modification, and with that, we've also completed Chapter 18 on biotechnology and genetic modification. Thank you so much for watching.